This slide is showing the organization of a skeletal muscle fiber, and here we're going to start to zoom in on a skeletal muscle fiber. So we begin with the skeletal muscle itself, and attaching that skeletal muscle to a bone is a tendon. When we look at the membrane surrounding the entire skeletal muscle, remember that is an epimysium. When we pull out individual fascicles, those fascicles are surrounded by a paramysium. And then within the fascicle are groups of muscle fibers, which are also the cells, the muscle cell itself. So surrounding each individual muscle fiber is an endoneurium, and deep to that is a plasma membrane called a sarcolemma. The entire structure within the sarcolemma are myofibrils, which have bundles of myofilaments. And these myofilaments specifically are going to be called myosin and actin, which we will be talking about more coming up. Some of the other structures shown in this group of myofibrils would be the structural unit of a skeletal muscle. And that's what's called a sarcomere. So that's a very important term to make sure that you know. And what we want to focus on now is the organization of one of these sarcomeres. So most of the volume of a skeletal muscle cell is composed of these cylindrical myofibrils that run the length of an entire cell. These myofibrils have the ability to shorten, resulting in contraction, and each of these myofibrils will contain myofilaments, which are the strands of proteins. And the two types of myofilaments are thick filaments, which are made up of myosin, and thin filaments, which are made up of the protein actin. So as I mentioned, the sarcomere is the structural and functional unit within a myofibril. A sarcomere is going to be made up of overlapping thick and thin filaments. And again, it's important for you to remember that the thick is the myosin protein and the thin is the actin protein. So one sarcomere is going to span from one Z disc to the next Z disc. So in order for shortening to actually occur, there's going to be a protein called troponin, which must bind to calcium. But again, what's going to be important here is to know the structure of the sarcomere, as we're going to see more and more in the next few slides. So the thick filament is the myosin, the thin filament is the actin. What happens is there's a cross bridge that is formed where the actin actually grabs the myosin and pulls it in a certain direction. So the microscopic anatomy of the sarcomere has very specific regions, which we actually see under the microscope. Those regions are going to be the thick and thin filaments, and it's important that you know these four regions. A little memory trick to remember these is the mnemonic hazy, and this will help you remember that the H zone is in the A band, and the Z disc is in the I band. So let's look at the I band first. It contains only thin filaments, but not thick filaments. The I band is bisected by the Z disc, so that's what it contains. So Z disc and I band, whereas the A band contains thick filaments, and because of that, under a microscope, it actually appears darker. This is going to account for that striated appearance that we see in a microscope slide. So the A band is located in the central part of the sarcomere, whereas the H zone is going to be in the center of the A band. Thus, we have the second part of our mnemonic. The H zone is in the A band. So hopefully that will help you to remember that the thick myosin fibers are present in the A band, but not the thin actin filaments. So this slide is showing us the structure of the sarcomere in a little bit more detail. If I zoom in here, we can see several different sarcomeres that are actually shown. So from this point to this point would be one sarcomere. From here to here would be one sarcomere. And from here to here would be one sarcomere as well. As we look on the right-hand side of the slide, we can see that the Z disc is in the I band. And notice that there's only going to be actin that's actually present in the I band because of the thin filaments. However, in the A band, you can see that it's much darker, and that would account for the striation, again, seen under the microscope. And that's because there are myosin myofilaments that are present. So again, these myofilaments are going to be the actin as well as the myosin. This is showing a different view of the sarcomere here, and you can further see the difference between the thin filament and the thick filament. So again, I'd like you to notice that the A band has the Z zone in it, so HA, HAZI. We have the Z disc in the I band, and you can see the proteins that are present. So just remember the thick filament is in the A band, which is going to be myosin. And in the I band, it has light striations. And that is because only actin is actually going to be present. And the actin would be this red protein here, which you can see a little bit of it in the A band, and then the rest of it is in the I band. So when we look at this structure under a microscope, this is a very nice looking image of a sarcomere. Again, the structural and functional unit of a skeletal muscle fiber. The H zone is in the A band, which again is going to be the thick filaments of myosin. Whereas in the Z disc is the very center of the I band. That's only going to be thin filaments, so it would be actin that is present. To look at another image of the anatomy of a sarcomere, I want you to notice the highlighted regions of the sarcomere. First of all, the one thing that we do see is this is going to be the Z disc right here. And remember, the Z disc marks the border of one sarcomere to another sarcomere. You don't see another Z disc over here, so we know this is really just showing the I band and the A band here. This structure here is the H zone, and the H zone is going to be the region that starts to disappear as the sarcomere contracts. So I want you to notice that the structure that is highlighted here, basically from here, including all of this, is going to be the A band. And remember, in the A band, that's going to be where the thick filament is. And you should know by now that the thick filament is going to be myosin. The Z disc is in an area where there's no striation, and specifically it would be the I band, which only has actin that's present. So in this slide, the highlighted blue area is the I band, where we only have actin. This is the H zone, where there is only myosin that is actually present in the A band.